In this video, we are going to discuss about the topic proteins and amino acids. So here is the topic: proteins and amino acids, chemical constituents of life. We are the basic structure. Proteins speak. We are the basic structure and function of life, composed of 20 amino acids. The building blocks organized into primary, secondary, tertiary, and quaternary structure, classified as simple, conjugated, and derived proteins. Proteins are the most abundant organic molecules of the living system. They occur in every part of the cell and constitute about 50% of the cellular dry weight. Proteins form the fundamental basis of the structure and function of life. Origin of the word protein. The term protein is derived from the Greek word protease, means holding the first place. Priscillus, Swedish chemist, suggested the name protein to the group of organic compounds that are utmost important of life. Mulder, the Dutch chemist, in 1838 used the term proteins for the higher molecular weight, nitrogen-rich, and most abundant substance present in animal and plants. Function of proteins. Proteins perform a great protein performs a great variety of specialized and essential functions in the living cell. These functions may be broadly grouped as a static, structural, and dynamic. The structural function: certain proteins perform brick and mortar roles and are primarily responsible for structure and strength of body. These includes collagen and elastin found in bone, matrix, vascular system, and other organs, and alpha keratin present in epidermis tissues. Dynamic functions. The dynamic functions of protein are most diversified in nature. These include protein acting as enzyme, hormones, blood clotting factors, immunoglobulins, membranes, receptor, storage protein, besides the function in genetic control, muscle concentration, respiration, and etc. Proteins performing dynamic function are appropriately regarded as the working horse of cell. Elemental composition of the proteins. Here is the elemental composition of the protein are predominantly constituted by 5 major elements in the following proportion: carbon 50 to 55%, hydrogen 6 to 7.3%, oxygen 19 to 24%, nitrogen 13 to 19%, sulfur 0 to 4%. Besides the above, proteins may also contain other elements such as phosphorus, iron, copper, manganese, magnesium, zinc, etc. The content of nitrogen as essential component of protein: on an average 16% estimation of nitrogen in the laboratory. Most is Zeldal method is also used to find out the amount of protein in biological fluids and foods. Protein are polymers of amino acid. Proteins are complete hydrolysis with concentrated HCl for several hours, yield L-alpha amino acids. This is the common property of all the proteins. Therefore, proteins are the polymers of levo-alpha amino acids. Standard amino acids, as many as 300 amino acids occur in nature. Of these, only 20 known the as standard amino acids are repeatedly found in the structure of protein, isolate from different form of life, animal, plants, and microbials. This is because of the universal nature of the genetic code available for the incorporation of 20 amino acids when the protein are synthesized in the cell. The process in turn is controlled by DNA, the genetic material of cell. After the synthesis of protein, some of the incorporated amino acids undergo modification to form their derivative. Amino acids are a group organic compound containing amino or carboxyl group. The amino group NH2 is basic while the carboxyl group is acidic in nature. General structure of amino acid. The amino acids are termed as alpha amino acids in both the carboxyl and amino groups are attached to the same carbon atom as depicted below. General structure exists as ion. The alpha carbon atom binds to the side chain represented by the R, which is different for each of the 20 amino acids found in protein. The amino acid mostly exist in the ionized form in the biological system. Optical isomers of amino acids. Optical isomer of amino acid. If a carbon atom is attached to 4 different groups, it is asymmetric and therefore exhibit optical isomerism. The amino acid except glycine possess 4 distinct groups RH, COO, NH3 held by alpha carbon. Thus, all the amino acids except glycine where RH have optical isomerism. The structure of Levo and dextro amino acids are written based on the configuration of levo and dextroglyceraldehyde. The protein are composed of L-alpha amino acid. Classification of amino acids. There are different ways to classify the amino acid based on the structure and chemical nature, nutritional requirement, metabolic fate. Amino acid classification based on the structure. A comprehensive classification of amino acid is based on their structure and chemical nature. Each amino acid is assigned a 3-letter or 1-letter symbol. The symbol are commonly used to present the amino acid in protein structure. The 20 amino acids found in protein are divided into 7 distinct groups. The different groups of amino acid, their symbol and structure are given. The salient features of different groups are described. The structural classification of levo alpha amino acid found in protein. Amino acid with aliphatic side chain. Glycine, Gly-G, alanine, La-A, valine, Val-V, branched chain, leucine, branched chain, isoleucine, branched chain, amino acid containing hydroxyl OH groups. sulfur containing amino acids. Cysteine is sulfahydryl. Cysteine disulfide. Methothione, thionine, thioether, aspartic acid, asparginine, glutamic acid, basic amino acids. These are the acidic amino acids and their amides. And these are the basic amino acids with their amides. Glycine, alpha amino, sigma amino, arginine, guanidino, histidine, imidazole, protein and amino acids. tyrosine, tryptophan, amino acid, proline, amino with aliphatic side chain. These are monocarboxylic acid. This group consists of the most simple amino acid glycine, alanine, valine, leucine, and isoleucine. The last three amino acids contain branched aliphatic side chain, hence they are referred to as branched chain amino acids. Hydroxyl group containing amino acid. Serine, threonine, and tyrosine are hydroxyl group containing amino acids. Hydroxyl group containing amino acid. Serine, threonine, and tyrosine are hydroxyl group containing amino acid. Tyrosine, being aromatic in nature, is usually considered under aromatic amino acid. Sulfur containing amino acid, cysteine with sulfur hydryl group and methionine with thioether group are the two amino acids incorporated during the course of protein synthesis. Cysteine, another important sulfur containing amino acid, is formed by condensation of two molecules of cysteine. Acidic amino acids and their amides. Aspartic acid and glutamic acid are dicarboxylic monoamino acid, while asparagine and glutamine are their respective amide derivatives. All these four amino acids possess distinct codons for their incorporation into proteins. Basic amino acids, the three amino acids lysine, arginine with guanidino groups and histidine with imidazole ring are dibasic monocarboxylic acid. They are highly basic in character. Aromatic amino acid, phenylalanine, tyrosine, and tryptophan with the indole ring are aromatic amino acid. Besides, these histidine may also be considered as this category. Amino acids. Proline containing pyrrolidine ring is unique amino acid. It has an amino group NH instead of an amino group. found in the another amino acid. Therefore, proline is an alpha amino acid. Heterocyclic amino acid, histidine, tryptophan, and proline. Classification of amino acids based on the polarity. Amino acids are classified into four groups based on their polarity. Polarity is an important for protein structure. Non-polar amino acids, these are amino acids are also referred to as hydrophobic, water hitting. They have no charge on the R group. The amino acid included in this group are alanine, leucine, leucine, valine, methionine, phenylalanine, tryptophan, and proline. Polar amino acid with no charge on R group. These amino acids such as carry no charge on the R group. They, however, possess groups such as hydroxyl, sulfahydryl, and amide and participate in hydrogen bonding of protein structure. The simple amino acid glycine, where RH is also considered in this category. The amino acids in this group are glycine, serine, threonine, cysteine, glutamine, asparagine, and tyrosine. Polar amino acids with group R group, with positive R group. Polar amino acid with positive R group. The three amino acid glycine, arginine, and histidine are included in this group. Polar amino acid with negative R group. The dicarboxylic monoamino acids, aspartic acids, and glutamic acids are considered in this group. Polar amino acid with negative R group. The dicarboxylic monoamino acid, aspartic acid, and glutamic acid are considered in this group. Nutritional classification of amino acids. The 20 amino acids are required for the synthesis of variety proteins. Besides other biological functions, however, all these 20 amino acids not be taken in the diet based on the nutritional requirement. Amino acids are grouped into two classes, essential and non-essential. Essential or indispensable amino acid. The amino acids which cannot be synthesized by the body and therefore need to be supplied through the diet are called essential amino acids. These are required for proper growth and maintenance of the individual. The 10 amino acids listed below are essential for human and also for rats. Arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan. The code AV, the first letter is the amino acids memorized to recall essential amino acids. Other useful codes are H, vital, LMP, PH, Villa, TT, PVT, TIM, HALL, MATT. The two amino acids, namely arginine and histidine, can be synthesized by adults and not by the children. Hence, these are considered semi-essential amino acids. Remember AH2 recall. Thus, 8 amino acids are absolutely essential, while 2 are semi-essential. Non-essential or dispensable amino acids. The body can synthesize about 10 amino acids to meet the biological needs. Hence, they need not to be consumed in the diet. These are glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, tyrosine, and pyrroline. Amino acids classification based on the metabolic fate. The carbon skeleton of amino acid can serve as a precursor for the synthesis of glucose, glycogenic or fat, ketogenic or both. From metabolic viewpoint, amino acids are divided into 3 groups. For details, glycogenic amino acids, these amino acids can serve as precursor for the formation of glucose or glycogen. Alanine, aspartate, glycine, methionine. Ketogenic amino acids can be synthesized from these amino acids. Two amino acids, leucine and lysine, exclusively ketogenic. Glycogenic and ketogenic amino acids. Glycogenic and ketogenic amino acid. The four amino acid isoleucine, phenylalanine, tryptophan, tyrosine are precursor for synthesis of glucose as well as fat. Selenocysteine, the 21st amino acid. As already 20 amino acids are commonly found in proteins. In recent years, a 21st amino acid, namely selenocysteine, has been added. It is found at the active site of certain enzyme protein, selenoproteins, glutathione, peroxidase, glycine reductase, 5-deionase, thyrodoxine reductase. Selenocysteine is an unusual amino acid containing the trace element selenium in place of the sulfur atom, cysteine. Incorporation of selenocysteine into the protein during translation is carried out by the codon, namely UGA. It is interesting to note that UGA is normally a stop codon and terminate protein biosynthesis. Another unique feature is the selenocysteine and enzymatically generated from serine directly on the tRNA, selenocysteine tRNA, and then incorporated into protein. Pyrosilane and 22nd amino acid. In the year 2002, some researchers have described that another amino acid, namely pyrrolysine, as the 22nd amino acid present in protein. The stop codon UAG can code the pyrosilane. Properties of amino acids. The amino acids differ in their physiochemical properties, which ultimately determine the characteristic of the proteins. Solubility, physical properties. Solubility, most of the amino acids are usually soluble in water and insoluble in inorganic solvents. Melting points. Amino acids generally melt at high temperature, often above 200 degrees centigrade. Taste. Amino acids may be sweet, tasteless, or bitter. Monosodium glutamate is used as a flavoring agent in food industry and Chinese food to increase taste and flavor. In some individual intolerant of NSC, Chinese restaurant syndrome is observed. Optical properties. All the amino acids except glycine possess optical isomer due to the presence of asymmetric carbon atom. Some amino acids also have a second asymmetric carbon, isoleucine, threonine. The structure of L and D amino acid comparison with the glyceraldehyde has been given. Amino acids as Ampholites. Amino acids as ampholites. Amino acids contain both acidic and basic group. They can donate a proton or accept a proton, hence amino acids are regarded as ampholites. The name Zwitter is derived from German word which means hybrid. Zwitter ion or dipolar is a hybrid molecule containing positive and negative ionic groups. The amino acid rarely exists in a neutral form with free carboxylic and free amino groups. In a strongly acidic pH, low pH, the amino acid is positively charged. It is negatively charged anion. Each amino acid has a characteristic pH at which it carries both positive and negative charges and exists as deuterion. Isoelectric pH. Isoelectric pH symbol Pl is defined as pH at which a molecule exists as a deuterion on dipolar ion and carries not net charge. Thus, the molecule is electrically neutral. The Pl value can be calculated by taking the average pKa value corresponding to the ionizable groups. For instance, leucine has two ionizable groups and its Pl can be calculated as follows. This is the structure of deuterion. The isoelectric pH. Existence of an amino acid as cation, anion, and deuterion. See the structure of the deuterion. Leucine exists as cation at pH below 6 and anion at pH below 6. At the isoelectric pH, leucine is found as deuterion. Thus, the pH of the medium determines the ionic nature of the amino acid. For the calculation of Pl of amino acids with more than two ionizable groups, the pKas for all the groups have to be taken into account. Titration of amino acids. The existence of different ionic forms of amino acids can be more easily understood by the titration curves. The graphic representation of leucinic titration is depicted. At low temperature, leucine exists in a fully protonated form as action. As the titration process with NaOH, leucine loses its protons and at isoelectric pH it becomes a zeta ion. Further titration results in the formation of anionic form of leucine. Some more details on isoelectric pH are discussed under the properties of protein. Chemical properties. The general reactions of amino acids are mostly due to the presence of the functional group or two functional groups. The group namely carboxyl and the amino group. Reaction due to the carboxylic group, amino acids form salt with bases and esters. With alcohol decarboxylation, amino acids undergo decarboxylation to produce corresponding amines. This reaction assumes significance in the living cell due to the formation of many biological important amines. These include histamine, tyramine, and gamma aminobutyric acid (GABA) from the amino acid histidine, tyrosine, and glutamate respectively. Reaction with ammonia. The carboxyl group of dicarboxylic amino acid reacts with NH3 to form amide. Aspartic acid NH3, asparagine, glutamic acid, glutamine. Reaction due to NH2 group. The amino group behave as bases and combine with acid to form salts. The amino groups behave as a basis. They combine with acidic to form salt. Reaction with ninhydrin. The alpha amino acid reacts in ninhydrin to form a purple, blue, or pink color complex. Ninhydrin reaction is effectively used for quantitative determination of amino acids and proteins. Fluorine and dihydroxyproline give yellow color with ninhydrin. Color reactions of amino acid. Amino acid can be identified by specific color, transamination, oxidation determination, non-standard amino acids. Beside the 20 standard amino acids present in the protein structure, there are several other amino acids which are biologically important. This includes the amino acid derivatives found in protein, non-protein amino acid performing specialized function of and the D amino acids.